Ribosomes. A ribosome is a complex molecule found inside the living cell that produce proteins from amino acids during the process protein synthesis or translation. Ribosomes, also called as ribonucleoprotein particles, assembly shop or protein factories. A ribosome is a complex cellular mechanism used to translate genetic code into chains of amino acids. A ribosome has a large and small subunit. How the ribosomes are different from other organelles. Ribosomes are different from other organelles. Ribosomes do not have membranes around which separate them from other organelles. Proteins are produced from ribosomes while floating in cytoplasm and later become membrane bound to the endoplasmic reticulum. Can a cell live without ribosome? Life is impossible without ribosomes. Ribosomes are responsible to synthesize proteins in living forms. Cells need protein to survive. Without protein, the cell would die. How can we compare the ribosome with a human life? Ribosomes are like a cement factory because cement holds things like bricks together, just like how ribosomes help to hold organelles together in a cell. We will learn about interesting facts of ribosomes. Ribosome is the smallest organelle in a cell. Ribosomes are the only cell organelles which can synthesize proteins and are also widely used in the production of antibiotic drugs by understanding its cellular mechanism. Ribosomes are produced inside the nucleolus of the nucleus. Ribosomes are not surrounded by a protective membrane. Do you know how ribosomes has originated and evolved? The evolution of the ribosome, a large molecular structure found in all prokaryotic and eukaryotic cells. Ribosome, cellular particle that is present in large numbers in all living cells and serves as the site of protein synthesis. Ribosomes occur both as free particles in prokaryotic and eukaryotic cells. Prokaryotic ribosomes evolved in 6 phases, sequentially acquiring capabilities for RNA folding, catalysis, subunit association, correlated evolution, decoding, energy-driven translocation, and surface proteinization. Who discovered ribosomes? Ribosomes was first observed by Albert Claude in 1941 and named it as microsomes. George E. Palade in 1955 discovered ribosomes and described as small particles in the cytoplasm that preferentially associated with the endoplasmic reticulum membrane. R.P. Robert in 1958 coined the term ribosome. Occurrence in prokaryotic cells. Ribosomes are microscopic factories found in all prokaryotic cells including bacteria. Occurrence in eukaryotic cells. Ribosomes are distributed throughout the eukaryotic plant and animal cells. Size and shape of ribosomes in prokaryotic and eukaryotic cells. Ribosomes appear flattened and spherical in shape when viewed under an electron microscope with a diameter ranging between 15 to 25 nanometer. Location in prokaryotic cells. In prokaryotic cells, the ribosomes often occur freely in the cytoplasm. Location in eukaryotic cells. In eukaryotic cells, the ribosomes occur either freely in the cytoplasm or remain attached to the outer surface of the membrane of endoplasmic reticulum. Chemical nature of ribosome. Ribosomes are composed of two subunits, the large and the small subunits. Both consist of ribosomal RNA molecules and a variable number of ribosomal proteins. Several fractal proteins catalyze different steps of protein synthesis by binding transiently to the ribosomes. Structure of ribosomes in prokaryotic cells. Prokaryotic ribosomes have two unequal subunits designated as small subunit and large subunit, depending upon their sedimentation coefficients. In prokaryotes, ribosomes include three separate rRNA molecules. Ribosomes consist of a large subunit and a small subunit. The proteins in ribosomes primarily help to fill in the structural gaps and enhance the ribosome's main role in the synthesis of proteins. Structure of ribosome in eukaryotic cells. Eukaryotic ribosomes are also known as ATS ribosomes, referring to their sedimentation coefficients in silver units, as the sedimentation rate is faster than the prokaryotic ribosomes. Eukaryotic ribosomes have two unequal subunits designated as small subunits and large subunit depending upon their sedimentation coefficients. Both subunits contain dozens of ribosomal proteins arranged on a scaffold and are composed of ribosomal RNA. The small subunit monitors the complementarity between tRNA anticodon and mRNA, while the large subunit catalyzes peptide bond formation. The ribosomes has three binding sites. A site carries a tRNA containing activated amino acid. B site carries tRNA containing polypeptide chain. E site exit site from where the deacylated tRNA is released into the cytosol. Types of ribosomes. Free ribosomes. Free ribosomes produce proteins that are necessary for the cell. Free ribosome includes proteins that are used for the metabolism of food. Free ribosomes synthesize proteins that are released into the cytosol and used within the cell. Bound ribosomes. Bound ribosomes produce proteins that are transported out of the cell. Bound ribosomes are located and attached onto the surface of the endoplasmic reticulum. The endoplasmic reticulum that contains ribosomes is described as the rough endoplasmic reticulum. Functions of ribosomes. Protein synthesis. During protein synthesis, the two subunits join together on the mRNA. Many ribosomes attach to the mRNA and forms a polyribosome. The ribosomes contain binding sites for the attachment of tRNA containing activated amino acids and tRNA containing peptide chain. The ribosomes move on the mRNA as they move along the triplet codon. The mRNA is translated and the peptide chain is elongated. The process of production of proteins. The deoxyribonucleic acid produces mRNA by the process of DNA transcription. The genetic message from the mRNA is translated into proteins during DNA translation. Disease caused due to defects of ribosomes in humans. Diamond blackfan anemia, 5q syndrome, Schwarzman-Diamond syndrome, cartilage hair hypoplasia.